Okay, I was walking by this model and decided to do a quick discussion on mitosis. And so I'm going to start over in the top left with interphase. So you know that the cell is in interphase in this part of the model because you see that the DNA is still in the form of chromatin. So chromatin and chromosomes are the same material, but they're just different forms. You know, the diffuse, very extended form of a chromosome is chromatin. When the cell is not dividing, it is in the form of chromatin, and then it will condense into chromosomes. So it's like how water can be a liquid or a solid. It's still the same material. So our DNA can be in the form of chromatin or chromosomes, still the same material. And so what goes on in interphase is the cell is preparing to divide. It spends about 90% of the cell cycle in interphase, going through gap 1, S, and gap 2. And then in those, it will grow in size, make organelles, and DNA for 2. So then you move over here and you see prophase. With prophase, the chromatin condenses into chromosomes. The nucleolus will disappear and the nuclear membrane is going to fragment and break apart. So over here, late prophase, maybe prometaphase, I would say that one and this one over here would be prometaphase-ish, into the prometaphase. And what's going on in prometaphase is you have spindle fibers are radiating from the centrosomes that are moving to the opposite poles. So those spindle fibers are microtubules coming out of the centrosomes. And so what's going to happen is they're going to attach to these structures called kinetochores. And kinetochores are these protein complexes on either side of the centromere. So the spindle fibers attach to the kinetochores, and then there's like this back and forth tug of war. This is, still prometaphase, this tug of war until metaphase, where now we have the sister chromatids lined up at the metaphase plate. So metaphase is about the stuff being lined up at the middle. And then anaphase, we can see they have 3 different parts here for anaphase. Early anaphase, later and latest, anaphase. And so with anaphase, the big thing here is sister chromatids become daughter chromosomes. So when they're still linked together, like here, they're still sister chromatids. Once they separate and start to go to opposite poles, now they're daughter chromosomes. Then here we get closer. They keep on going to opposite poles. Notice how cytokinesis is going on during anaphase. So you can see the cleavage furrow starting to form and the cleavage furrow getting bigger because cleavage is how animal cells are going to divide. So cytokinesis is proceeding during anaphase. And then telophase is just the opposite of prophase. We reform the nuclei, the chromosomes will start to go back into chromatin, the nucleolus is going to reform, and then we continue the process of cytokinesis till we have 2 new daughter cells. So just to reiterate, interphase, cells getting ready to divide, 90% of the life cycle. The grainy chromatin and the intact nucleolus are going to be keys to identifying that. Then we have prophase, chromatin becomes chromosomes. Here we see over here, later prophase, prometaphase, centrosomes are migrating to opposite poles with the spindle apparatus, these microtubule fibers coming out of it, attaching to kinetochores, tug of war, line up in metaphase at the metaphase plate. And then we have anaphase, where the sister chromatids become daughter chromosomes. And then we have prophase. We reform the nucleus and we complete cytokinesis during prophase. So that is the basics of mitosis in an animal cell.